Hello, everyone. My name is Maria Lopez. I am currently in my second to last semester of the University of Florida's Master of Public Health program. My concentration is social and behavioral studies. Therefore, I chose my project to be regarding mental health, specifically mental illnesses. To connect mental illness with environmental health, I chose my topic to be toxin exposure during childhood and mental illness development. In the next few slides, I will demonstrate data found from previous studies and in literature to support this claim. So what are toxins? Toxins are poisonous substances derived from living organisms that are notably toxic when introduced to tissues and typically are capable of inducing antibody formation. Three major toxin sources found in the environment are bacterial toxins, toxins from fungi, and toxins from algae and plants. In this presentation, we will explore how these toxins affect not only our physical health, but our mental health as well during the developmental period. This class focuses not only on environmental health for human beings, but involves the One Health approach in order to understand how human health, plant health, and animal health are all connected and how maintaining optimal health in all three realms is important to ultimately achieve optimal environmental health. So for this topic, I will be making the One Health connection by explaining how firstly, plant health is affected by this public health crisis. Plants produce toxins, therefore they are the producing factor of this crisis. And in order to avoid adverse health effects, both humans and animals must avoid contact and consumption of these plants that produce toxins. Animals who consume these toxins producing plants have shown adverse physical and mental health effects and affect us humans as we consume the meat of these animals that consume the toxin producing plants. Like the animals, in order to avoid intoxication, we must avoid eating the meat from the intoxicated animals in order to avoid adverse physical and mental health effects. The method used to conduct this research study is by using secondary data analysis along with literature review and health communication data review methods. Secondary data analysis and literature review will allow me to find already existing research regarding toxin exposure and mental illness development and analyze whether the data found either supports my claims, does not support, or if I find data that both supports and does not, which, spoiler alert, is what you will find in the case of this study. Health communication data review essentially will be conducted by searching for any previous or active health campaigns that have been brought about to support my claims of toxin exposure and mental health effects. So first, I searched data and literature from the UF library database. Then I searched data and literature from external sources such as Google Scholar and PubMed. My findings were really interesting as I found both supportive data and contradictory data. So we had spoken about all the kind of different toxins and how some come from bacteria, plants, algae, and fungi. Most of the data I found regarding the effects towards mental health, most of it was regarding bacterial toxins. So much of this study focuses on bacterial toxins. Much data was found in both the internal and external databases regarding the microbiota gut-brain, or MGB. The microbiota gut-brain is defined as a network of connections involving the brain and the digestive system. Here you will see an illustration of what the MGB looks like in the first picture of the slide. In this second picture, what is shown is an illustration of the different methods that MGB is manipulated for research purposes. These manipulation methods include the introduction of antibiotics, which promote the reduction of bacterial infections, probiotics, which are live microorganisms that serve to promote healthy gut flora, prebiotics, which are plant fibers who serve the same purpose as probiotics to promote healthy gut flora, the introduction of germ-free mice, which are mice who have lived in a sterile environment their whole life, therefore are not prone to bacterial infections, and phage therapy, which introduces phages or viruses that infect unwanted bacteria. This is mostly used when antibiotics are unsuccessful. There is FMT, which is fecal microbiota transplantation, which involves the transfer of fecal matter from one individual's gut to another. And lastly, there is polybiotics, symbiotics, and psychobiotics. Polybiotics is the introduction of a combination of bacteria. Symbiotics are the combination of pre and probiotics. And psychobiotics are those probiotics, prebiotics, and symbiotics that have shown a mental health benefit. All of these manipulation methods have been found to be successful in helping explain ways that proper nutrition and lack thereof affect the microbiome of our digestive system and in turn our brain, especially amongst expectant mothers and how their nutrition affects the development of their unborn children. Studies using MGB manipulation methods have proven supportive evidence towards differential microbial gut composition amongst patients diagnosed with major depressive disorder, bipolar disorder, anxiety disorders, schizophrenia and psychosis, ASD, autism spectrum disorder, attention deficit disorder, and neurodegenerative disorders such as Parkinson's disease and Alzheimer's disease. In the last slide, you can see results of a study that involved introduction of fecal matter transplant, FMT, in order to see the difference in behaviors from mice who were diabetic to those who were not diabetic. This study found a significant difference in avoidant and depressive behaviors amongst diabetic mice and non-diabetic mice. And once a transplant occurred, those non-diabetic mice who received the transplant showed the same behavioral, avoidant, and depressive responses as the diabetic mice. Now, contrary to my claims, much data was found regarding the actual benefits of toxin exposure towards neurological and mental health. One bacterial toxin, a botulinum toxin, has actually been found to aid in spasticity, dystonia, facial spasms, tremors, Tourette syndrome, headaches, and pain relief. This botulinum toxin, or BONTA, as it is clinically known, is an approved treatment for cerebral palsy amongst children. Now, when I was searching for data on health communication campaigns, a few stood out to me that were important to highlight. The first one is an advertisement for a kombucha brand, Health Aid, which promotes the beverage to help maintain a good gut feeling, showing someone eating nachos and drinking kombucha to maintain a balanced gut. Kombucha is a fermented drink that contains a symbiotic culture of bacteria and yeast, which has been found to help balance gut health and mental health. Another campaign I found interesting was from Kellogg's, the cereal brand, which made an interactive virtual reality campaign named Gut Bacteria Reef, which shows kids the importance in taking care of your gut, showing the similarities of a coral reef to your gut, which consists of trillions of organisms that needs nutrients to thrive, promoting the consumption of their cereal, which contains high fiber, which is helpful in maintaining gut health. The next two images are part of a campaign from Mental Health America called #4Body4Mind Challenge. This challenge was introduced this past May to promote healthy physical health and mental health strategies one can implement into their daily lives. The second image shows a shot of a short educational video explaining the gut-brain connection, as we spoke about earlier. The third image shows a mental health grocery list, which includes many whole nutritious foods, including prebiotic foods as well as probiotic foods to promote gut health and mental health. This campaign is great as it promotes living a healthy lifestyle among all walks of life, not just for adults, but growing children as well as expectant mothers. In all, this research allowed me to explore how toxin exposure can truly affect mental health and how lack of nutritional value during mealtime can result in poor mental health during the developmental stage more than any other stage, as it is the most vulnerable stage during the human life cycle. It also provided me with insight to some benefits in toxin exposure that have already used as effective treatment for neurological disorders. In the future, my hope would be that there will be treatment to promote gut healing that can be used in combination with psychotropic medication as needed in order to promote overall wellness in both body and mind. Thank you for being here, and I hope you enjoyed my presentation.